Welcome, students. Great to be with you again. And in this brief presentation, I'm going to talk about how information is copied from one cell to another. This is the process we call DNA replication. I hope that you have your PowerPoint notes with you. We're going to go over this brief presentation, at the end of which I am going to allow you to pause and watch a video that I have posted elsewhere. And I'll tell you more about that in a moment. All right, so here we go. If you could look at particularly prepared sections of cells through their different phases, you may be familiar with this process if you've taken a biology or a genetics course. Cells are typically containing their DNA in a structure that we call chromatin. We talked about it in the previous lecture. As the cell prepares to divide, all of these particles of DNA and the histone protein complexes, start condensing into structures that we call chromosomes. Eventually, they line up in the middle of the dividing cell, and as the cell divides, a copy of each set of chromosomes moves away and eventually is encased in a new cell. Now, let's look at what is happening at the actual molecular level with the DNA itself. So when DNA is replicated, remember that DNA consists of a double-stranded helical structure, right? And each strand of the double helix is now going to serve as a template for the manufacture of a new strand of DNA. In other words, each strand of the double helix is going to be copied into a complementary strand of itself. This is a schematic here. As you can see on the top, you see the double helix. Notice that as you reach the middle of the picture, it starts unwinding. And as it does, a new strand is being copied of each original or template strand. In each of the daughter DNA strands, one strand is from the parent DNA. This is the one here in a light yellow color. And the other one is the new strand, which is in a darker kind of brown color. That would be the daughter strand. So as you can see, the new DNA sets are going to have one strand that was part of the original and one strand that is a daughter strand. This is a phenomenon that we call semi-conservative replication. Semi-conservative, because we conserve one of the original strands, that's a template, and the daughter strand is new. Here is a schematic of some of the things that are involved, and there are going to be different enzymes involved in the process. Remember, the original structure of DNA consists of the double helix. In order to replicate it, we had to first unwind it and separate them. And you're going to have different kinds of proteins stick to it. For example, you're going to have some single strand binding proteins. These are proteins that are meant to stabilize the single stranded form of the DNA. You're going to have an enzyme called DNA polymerase, which is going to be in charge of directing the copying of each parent strand into a daughter strand. And there's going to be an advance of this loop as we go from one direction of the DNA to another one. We call this the replication fork. As you can see in the bottom here, let me blow this up. Let's assume that you have a template DNA strand. Here is on the right side. Notice that right now I'm showing it with the 5' end of that strand in the bottom and the 3' end at the top. This template strand is going to be copied onto a daughter strand. Now the daughter strand is going to be assembled beginning at the 5' end of that chain and growing. So what's going to happen is, let's say you have a strand that's growing here, and this is the last nucleotide that was added. Notice that, again, the 2' carbon of the ribose ring does not have an OH because this is deoxyribose. Notice that we have a 3 hydroxyl group at the 3' end. Now, the incoming base that's going to be added is going to come in the form of what we call a nucleoside triphosphate. So this is a base attached to a ribose ring, which at the 5' end has a triple or triphosphate group. The OH from the 3' end of the growing chain is going to connect to this phosphorus atom here. And as it does, this phosphorus oxygen bond will break and release this whole double phosphate group that we have talked about before. We call it the pyrophosphate. And as a result, the new nucleotide that was brought in gets connected to the 3' end of the growing strand and is available at its 3' end to continue. So you can see here in this case, for example, let's say that your parent DNA had a sequence here going 5' A, C, T. The growing strand is coming in the anti-parallel direction, and they're going to attach bases that are complementary to the ones on the template. So for example, if the template here had a T, then the complementary one has an A. If the template strand had a C, the complementary one is going to have a G. If the template had an A here, now it's going to add a T, and here is the incoming TTP, in other words, thymidine triphosphate, and it's going to connect to the 3' end of the growing strand. This is essentially the basic biochemical step that goes on. Understand that in the process, there is this enzyme called DNA polymerase and other enzymes that are going to be involved. Here is a schematic on the left, and you can see how the DNA double helix is getting unwound here around the middle. And here you have the enzyme complexes that are moving along the DNA strand to unwind the double helix, disrupt the hydrogen bonding between the bases, and therefore exposing them so that the new strands can be added. Over here you have a schematic of one replication loop, and as you can see, as the enzymes are moving, let's say in direction in this picture from top to bottom, you can see how bases are being added one at a time, complementary to whatever was present on the parent or template strand. In this picture, the little purple T-shaped things are the nucleotides that are getting added. The purple strand here represents the daughter strand, and the yellowish one or beige one represents the parent or template strand that is being copied to get this. At the end, you're going to end up with two replicas of the original double helical DNA, but now you have two of them. One strand of each represents the original parental or template strand, and in purple you can see the daughter strand that has been replicated. And the idea is that in this manner you generate exact replicas of the original DNA sequence. At this point, I'm going to ask you to please maybe pause the video and go to your Canvas page, in which I have this video that shows you in an animation the whole process of DNA replication. I do not have it in here because of copyright issues, so please make sure that you go to the corresponding page in our Canvas site and watch this video called DNA replication, and then please come back. Okay, I hope that you have seen that video and you're back here to our original one. So here is again a duplicate of what you just saw in just one, let's call it a splash scene or a screenshot. And you can see in here all of the different components. Remember, as the video shows, you have some enzymes that I call sometimes helicases or topoisomerases that are basically unzipping the double helix. As the original single strands of template DNA separate, you're going to have single strand binding proteins that are going to stabilize that and keep it safe from other enzymes that are in the habit of cutting single stranded DNA. They're called nucleases. Now, there are going to be two strands. One of these strands is already completely open, so that as the DNA double helix unwinds, the DNA polymerase enzyme can simply continuously replicate that strand into a new one that is now coming in the 5' to 3' direction. On the other side, off of the other template strand, you cannot have the same thing happen because this strand is already in the 5', 3' direction in this way, going from right to left. And so you had to replicate it in segments or in fragments. And as the video showed us, the first thing that happens is at any given point, an enzyme called a primase attaches and makes a little RNA short-stranded primer, to which DNA polymerase adds a sequence of DNA. Eventually, what's going to happen is that you're going to form these different fragments. They're called Okazaki fragments because this is the author who discovered these things. So basically, what's going to happen is you're going to have all these fragments, which eventually have to be joined together by an enzyme called the DNA ligase. Also, the original RNA primer has to be kind of cut up by a different type of enzyme. And then once it's done, it gets filled in by the DNA polymerase. And therefore, afterwards, you'll have a chance to cut it up. To join these segments. So again, one strand of DNA is being replicated continuously. The other one is being synthesized or replicated discontinuously into small fragments that then have to be joined up to form the complete daughter strand and therefore the complete double-stranded, double helical daughter DNA. Now, as the process is happening, there's an interesting phenomenon, and it is that technically about once every 10,000 times, the wrong residue is inserted. So where there should have been an A G gets inserted. Where there should have been a T, a C gets inserted. Something like that along those lines. Now, that would lead to potential problems because then you would not keep the integrity of the original sequence of DNA. So what we have is we have several mechanisms of proofreading and repair. First of all, DNA polymerase itself kind of proofreads its work. In other words, it replicates a sequence and then it goes back and checks and makes sure that all of the base pairs are aligned correctly. If there is a mistake, the DNA polymerase will clip that residue and restart the sequence replication. Let's say that the proofreading does not catch the error and it tries to go through. Well, there's another set of DNA repair enzymes that will monitor the new DNA double helix, and they'll be able to catch the problems. They'll see where there is set, for example, a set of base pairs that don't match. And so it'll catch it, and what it'll do is it'll clip the DNA strand in there and replace it and fix it. So between the proofreading work of the DNA polymerase and the work of these other DNA repair enzymes, you can improve the rate during replication from 1 every 10,000 times to maybe like 1 in every billion bases. And so it helps to assure that the DNA strands that you've replicated have the correct sequence that they're supposed to have, and so they will pass that sequence of DNA to the next generation of cells as the cells themselves duplicate. Okay, in our next lecture, we're going to talk about the other important nucleic acid, which is RNA. So thank you for your attention. Again, please go back to our Canvas page that has the lecture notes for this topic, and make sure you catch that video about DNA replication. Thank you very much.